I'd like to talk about how microglial polarization is affecting the neuropsychiatric symptoms of the tuberous sclerosis complex mice model. I'd like to start by talking about the tuberous sclerosis complex, TSC. TSC is one of the neurocutaneous syndromes and genetic disease. The causative genes are TSC1 and TSC2. In TSC, many hamartomas and nodules are formed all over the body. In addition, epilepsy and multi-psychiatric symptoms also occur at a high rate. These neuropsychiatric symptoms are called TSC-associated neuropsychiatric disorders, TAND. It's so that TSC symptoms are caused by hyperactivity of mTOR complex 1, mTOR1. This mTOR1 hyperactivation occurred by the mutation in TSC1 and TSC2 gene. mTOR exists as two large protein complexes. One is mTOR complex 1, mTOR1, and the other is mTOR complex 2, mTOR2. And in normal cells, TSC1 and TSC2 replace and regulate mTOR complex 1, mTOR activity. But in TSC, TSC1 and TSC2 do not function, so mTOR becomes overactive. mTOR inhibitor Sirolimus suppress the mTOR activity. Therefore, Sirolimus improves some symptoms in TSC. Sirolimus is known to be able to suppress hamartomas in TSC, but the effect of Sirolimus on TAND is not yet clear. We have already created TSC2 conditional knockout mice with MIT Koike mice. We call these mice TSC2 secure mice, and we found that these mice exhibited spontaneous epilepsy. Therefore, we analyzed whether these TSC2 secure mice show TAND symptoms, and then we found one of the causes of TAND and epilepsy. First, we analyzed whether these TSC2 secure mice have the same phenotypes as TAND. First of all, we investigated about seizures in more detail. During 5 to 6 weeks age, we injected mice with Sirolimus or Makiko for 7 days, and we counted seizures in these mice with treated with Sirolimus or Makiko. This is one of the common spontaneous seizures seen in these secure mice. This graph shows the degree of the seizures. Here is secure mice with Makiko and with Sirolimus. Secure mice exhibited many and strong seizures, but Sirolimus treatment suppressed the seizures in secure mice. Next, we wanted to know whether TSC2 secure mice exhibit TAND symptoms, so we conducted behavioral analysis after Sirolimus and Makiko treatment. The first analysis was using the open field test. We looked into anxiety in mice. The stronger the anxiety, the longer the mouse spent in the broader area of the open field, this green area. In this graph, the higher values, the more anxiety. Secure mice have strong anxiety, but after Sirolimus treatment here, anxiety in secure mice was reduced. Next, in this light graph, we analyze mouse hyperactivity from velocity. Quicker movement typically indicates hyperactivity, and hyperactivity is one of the characteristics of ADHD. In the secure mice, it shows high velocity, but in Sirolimus-treated secure mice, the velocity was decreased to the same level as control mice. In summary, secure mice exhibited anxiety and ADHD-like behaviors, and those phenotypes had improved by Sirolimus treatment. Next, we used 8-armed radial maze to investigate about spatial working memory. In this test, we placed a food pellet as the tip of each arm here. Let the mouse learn the location of the food. We counted one error if the mouse enters the arm once visited. If the mouse has many errors, the spatial working memory is considered defective. This graph shows number of errors. The secure mice had predominantly higher error counts. However, Sirolimus-treated secure mice had reduced to number of errors. This result indicates that TSC2 secure mice are defective in spatial working memory, and that these defects are improved with Sirolimus treatment. Next, we evaluated the social behavior. In this social interaction test, we put 2 mice in the same cage. They are unfamiliar mice. We examine the time when mouse approached another mouse from itself. In the graph, shorter times indicate less sociability and autistic tendencies. The secure mice is in red had very short approach times. That is, it exhibited autism-like behavior. In blue is the Sirolimus-treated secure mice, and Sirolimus treatment did not change this to autism behavior. To summarize so far, TSC2 secure mice indicate multiple neuropsychiatric symptoms, and these phenotypes are improved by Sirolimus treatment, except for autism-like behaviors. Next, we investigated the causes of the epilepsy and turn in TSC2 secure mice. Immunostain analysis showed that the microglial morphology of Sirolimus-treated secure mice was significantly changed. In the upper panel, red indicates microglia, and the bottom panel, the microglial branches are traced. In Sirolimus-treated secure mice, microglial size was much larger than that of the other groups, and the number of branches in microglia was significantly higher than that of the other groups. Therefore, next we investigated the cause of changes in microglia by Sirolimus treatment. Microglia can be categorized in 2 main properties: pro-inflammatory M1 or anti-inflammatory M2. These polarization states change depending on the surroundings. Some marker genes are known to be expressed in each state. So we analyze the expression of M1 and M2 marker genes in microglia by using quantitative RT-PCR. In this graph, right gray bar is CQ with Makiko, and white one is CQ with Sirolimus. M1 marker, IL-1 and IL-1 beta tend to be high expression in CK mice, this flavor. While the M2 markers were predominantly elevated in Sirolimus treated CK mice, this white bar. Furthermore, we investigated the expression of cholinergic receptors in microglia because the microglial morphology and functions are depending on the expression of cholinergic receptors. These are some cholinergic receptors. Among these cholinergic receptors, PTX7, PTX4, and A2A receptors are commonly expressed in M1 microglia. While the P2Y12 receptor is reported to be expressed on M2 state microglia and to be involved in the microglial process extension. In contrast, A2A receptor is involved in the microglial process retroaction. Look at this graph. Right gray bar is CK with Makiko, and the white one is CK with Sirolimus. The expression P2Y12 receptor was significantly increased in Sirolimus treated CK mice. As I already pointed out, in Sirolimus treated CK mice, the branch number of microglia are increased. Then look at the graph below. In CK mice, this white gray bar, microglia expressed predominantly higher levels of A2A receptor, while expression of both PTX7 and PTX4 receptor were not significantly higher. These results suggested that in CK mice, microglia probably have less intense inflammatory properties, and then Sirolimus treatment makes them anti-inflammatory. Next, we measure the levels of ATP and ADP in CK mice brain. Because the ligand for P2Y12 receptor is ADP. Look at this graph. More surprisingly, ADP level in Sirolimus treated CK mice was 6 times higher than the other treatment. We consider the possibility that ADP increased level led to the activation of P2Y12 receptor, resulting in increased branching of microglial projections. Additionally, we also investigated the expression of ectonucleotidases. These ectonucleotidases. Among ectonucleotidases, we focused on LTP-2. LTP-2, ATP, but little ADP. Ectonucleotidases catalyze ATP into AMP, ATP into ADP, AMP, and adenosine. But LTP-2 degrade ATP, but little ADP. In this graph, white bar is Sirolimus treated CK mice, and LTP-2 expression was predominantly elevated in Sirolimus treated CK mice. To sum up, The TC2CQ mice were exhibiting symptoms as stunned. These stunned symptoms are improved by Sirolimus treatment, but not odin-like behaviors. In TSC-CQ mice, microglial showed elevated expression MR marker and HCl receptor. After Sirolimus treatment, microglia had increased expression M2 marker, P2I12 receptor, and NAPDS2. As a result, the microglial morphology changed. These results suggested that microglia in CQ mice are close to the pro-inflammatory state, and that Sirolimus treatment shift microglia to anti-inflammatory M2 state, and M2 state microglia may be protective for the nervous system. This research was supported by these individuals and many facilities staff in Osaka University. I'd like to express my sincere gratitude to those who supported this research. That's all I have to say. Thank you very much for your kind attention.